Hello students, welcome to the biology class. So in the last class we learned about the post-fertilization events, that how the zygote starts dividing, and as it divides it slowly moves through the fallopian tube. It starts moving from the ampullary isthmus junction, then it passes through the whole length of the isthmus, that's the longest part of the fallopian tube, then it enters into the uterus, and at the right place it will undergo implantation. We understood about the role of zona pellucida, that how it does not allow the wrong place of implantation. So because the trophoblast cells or trophoectodermal cells are phagocytic in nature, they can easily cause the release of enzymes and cause the breakdown of endometrium and can go for a wrong place of implantation. So to stop that, zona pellucida remains around the zygote as it's continue to divide. So in the last class we learned all these things, that zygote divides into 2 cell, then finally 4 cell stage, and then 8 to 16 cell stage. So the initial cell stages are called as the blastomeres, and the division is called as cleavage. If you remember, cleavage does not involve a long interface, or you can say there is hardly any growth in the size of the cell. The cell only prepares the raw materials required for the next division during a very small interface. And then the blastomeres change into 8 to 16 cell situation, which is called as morula. Morula can even extend up to 32 cell stage, and finally it turns into a blastocyst. Blastocysts carry 64 cells. In the morula, generally all the cells are similar, surrounded by a zona pellucida, but when you see the blastocyst, by the 6th to 7th day blastocyst is formed and it undergoes implantation by the time. So a blastocyst is made up of an outer layer of trophoblast, and the inner, at one corner you will see the inner cell mass. Inner cell mass gives rises to the embryo, and the trophoblast develops the placentation or helps in the implantation or attachment. Then we learned that by the 8th day trophoblast develops into 2 layers. Inner is cytotrophoblast and the outer one is the syncytio. Syncytio means multinucleate, and this helps in the formation of the chorionic villi or trophoblastic villi. So finger-like projections will develop. Rest all was regarding the gestulation, which is missing from the NCERT book. So I don't feel like summarizing that concept again. Now the main thing to understand here is that there are 4 extra embryonic membranes or structures which develop during the embryogeny, or during the formation of the embryo, or during the time of the pregnancy. So those structures are yolk sac, chorion, allantois and amnion. I have underlined those, highlighted those main names. So let's understand the function of these fetal membranes. So the first one is the chorion. So chorion is this one. Chorion is it's the outermost. You can see in this picture, this is the embryo drawn. So this is the outermost layer which surrounds the whole structure. So chorion is the outermost fetal membrane, which is formed by the outer layer of trophoblast, and it forms villi. I think you can easily see there are structures which I have drawn, like extension type of thing or villi type of thing. So all these structures are the villi. So the chorion forms the villi and the other fetal parts of the placenta. So all these things are done by chorion. Now the function of chorion is to form the fetal portion of placenta, because if you learn about various types, I'll be teaching you that topic today. Placenta has an important role of chorion in its formation. So that's why chorion's main function is to form a part of placenta. It also provides attachment to the fetus. Now because it forms the placenta, it helps in obtaining nutrition and then release of the excretory material. So all these things are possible, and gaseous exchange. So everything is possible through the structure called as chorion. Next to the chorion, the next one is the amnion. Now you can see this embryo is surrounded by a cavity. This cavity is filled with amniotic fluid, and the layer which surrounds it is called as the amnion. So what is amnion? It is the inner fetal membrane which lies immediately. So it is just outside the embryo. It lies immediately around the embryo. The fetal membrane is formed by the inner layer of trophoblast. it encloses a cavity. That cavity is called as the amniotic cavity, and there is a fluid filled there which is called as the amniotic fluid. Now that fluid will definitely have some dead floating cells, because the embryo is developing inside this cavity. So there are cells of the shed cells of the skin of the embryo inside, so they can be collected and they can be examined to understand if there are any genetic disorders in the child, so that abortion could be practiced. Then amnions acts like a shock absorber also, because there is a liquid inside this cavity. So liquid acts like a shock absorber, regulates the pressure, regulates the temperature. It also helps the child, the embryo, not to undergo desiccation. In the last class I told you desiccation means to dry out. Then it also helps in the growth and the movement of fetus. So all these are the functions of the amnion. After amnion, you can see next to the chorion there is one more portion or a cavity which is called as the allantois. So what is allantois? It's a membrane lined small sac-like structure. You can see there is a cavity I have drawn here. Sac-like